Our immune system comprises of a number of cells which protect us from microbial invasion. It can be broadly subdivided into innate immunity and adaptive immunity. Let us look at this in more detail and understand pidotimod, a multifaceted molecule which acts to stimulate both the innate and adaptive immune responses, thus protecting us from different microbial invasions. Let's see how pidotimod affects our innate immunity responses. Pathogens usually enter our bodies through various orifices. It can also enter through wounds that breach the skin barrier. Macrophages, neutrophils, and dendritic cells play a very important role in our innate immunity system. They recognize the pathogen as an antigen through PAMPs and bind to them with the help of toll-like receptors or TLRs. Pidotimod upregulates the expression of TLR2 on innate immunity cells. After antigen TLR binding, these cells engulf the pathogen in phagosomes. Phagosomes merge with lysosomes to form phagolysosome, breaking down the antigen and releasing the soluble debris by exocytosis. Pidotimod increases chemotaxis and phagocytosis by macrophages and neutrophils. After phagocytosis, macrophages and dendritic cells express a part of the antigen on their surface with a MHC2 receptor and act as antigen-presenting cells to the T cells in the lymph nodes. Pidotimod induces dendritic cell maturation. Let's see how pidotimod stimulates our adaptive humoral immunity. The antigen-presenting cells, or APCs, present the antigen on its surface with the MHC2 receptor to the naive CD4 T cells. The naive CD4 T cells get activated to form CD4 T cells or T helper cells. Pidotimod upregulates expression of HLA-DR. The dendritic cells also have ICAM1 receptor that binds with LFA1 receptor, facilitating antigen receptor binding. Pidotimod has important effects on ICAM1. Once activated, they stimulate B cells. Th1 immunity stimulates IgM, IgG, and IgA production, leading to macrophage activation. This is promoted by IF gamma release. Th2 immunity stimulates IgE production. Pidotimod stimulates IF gamma release, upregulates CD3 protein on T helper cells, corrects the Th1-Th2 balance, and also increases sputum IgA. Let's see how pidotimod stimulates our adaptive cell-mediated immunity. All human cells express the MHC1 receptor, which can present intracellular antigens to naive CD8 T cells. Once activated, these cells secrete cytokines such as IL-2. This activates cytotoxic T cells or T killer cells. These cells destroy the abnormal or infected cell by surrounding it in the form of a rosette. Pidotimod stimulates cytokines IL-2 release and rosette formation of T killer cells. Pidotimod and NK cells. NK cells are unique as they have the ability to recognize the pathogen in the absence of MHC1 self-marker. They destroy the abnormal cells by the release of perforins, which is a cytolytic protein. Pidotimod enhances NK cell function. Hence, pidotimod stimulates both the immunological mechanisms involved in humoral and cell-mediated immunity. In innate immunity, pidotimod upregulates TLR2, increases chemotaxis and phagocytosis of neutrophil and macrophages, induces DC maturation, and enhances NK cell activity. In adaptive immunity, it upregulates HLA-DR, affects ICAM1, stimulates IF gamma release, corrects Th1-Th2 balance, upregulates CD3 protein on T helper cells, increases sputum IgA, and stimulates IL-2 release and rosette formation of T killer cells.